Hello. We know that change is the only constant. Everything in the world and universe keeps changing. In fact, the universe is continuously expanding, and thereby that is itself change. The same way, the genome also changes. There is constant change in the genome. That is why we have so many diverse organisms in this world. I'm Dr. BCMR. We'll have a discussion about the nature of changes that happen in the genome. So the change in biology, we call it as mutation. And mutation is a result of or initiated as damage. Not only damage, but there are also other ways of mutation occurring. And there are consequences because of mutations. There will be a lot of terminology that we'll have to pick up while we try to learn a little bit about the mutations. Say, for example, I have a sequence here that is shown here, which we will consider as wild type or normal. And then we have had a change here. That is, the C has changed to A. And that is, C is substituted by A. So we call those as substitution mutations. In other ways, a G is added into between, say, TAC. Between these two AC, we have a guanine added, and that is an insertion mutation. Similarly, we can also have a deletion mutation where we had TAC was there. In this case, the C is removed, so now we have a different sequence. That is a deletion mutation. We can also have an inversion mutation, duplication mutation as well. Say, for example, this is an inversion mutation that is shown. The original sequence was something like GATAC, GAC, TTG. Now we have this TAC, GAC is inverted. So we have CAG, CAT. So that is an inversion mutation. We can also have duplication. In this case, the region TACG is duplicated again, TACG. What we have shown here are point mutations. That means they happen at a very small stretch, and those are point mutations. The same thing, something like deletion, insertion, or inversion duplication can happen even in larger scale. Could be 100 base pairs or more also. But when the mutations are small changes in one basis, we call them as point mutations. Then all the mutations or majority of the mutations start as DNA damage. Damage can be many types. Say, for example, we may have a nick in the sense we might have a small, the phosphodiester backbone, the phosphate backbone that we have on the DNA could have a base cut here like that. That is a mutation as well. That is not a mutation, but it is a kind of damage, which can easily normally be sealed by ligase. There could also be sometimes some of the bases, there might be a cleavage in the glycosidic bond because of which a base might be missing. You call those sites as apurinic or apyrimidic sites, or generally a basic site. So we have also several others, like not just the whole base missing or nitrogenous base missing. We can have functional groups removed. Say, for example, N6, the amino group on the 6th carbon could be deaminated or removed, lost, which is then it's just purine there. And those are called, those kinds of damages are called deamination. And some of them we may have addition of ethyl or methyl groups. Those are called as alkylation. And sometimes some of the functional groups of nitrogenous bases could be oxidized, that is oxidation. And because of radiation, sometimes you may have adduct formed. Those are something similar to that of thymidine dimers. So these damages contribute to the mutations or changes in the sequence itself. So that is the first primary definition of a mutation. Any change in the sequence is a mutation. So mutations will happen by, at least one way is by damage. Damage could be spontaneous or induced, and it can also happen spontaneously because of replication errors or because of hydrolytic damages, just like that too. So these mutations can be many ways, and what we will see is the consequences. What kind of consequences would they have if a mutation occurs? That all depends on where the mutation occurred and where the mutation occurred, what mutation it is, and how much mutation has happened, how big the fragment is. So to start with, here we have a promoter and an open reading frame, the broken arrow indicating the promoter. If there is a mutation indicated by this red spot here, then the promoter is malfunctioned, or assume that it is gone or it's no longer functional, because of which there might be no transcription at all. And if a mutation happens between the intergenic region, say this is open reading frame and this is the downstream open reading frame, and if insertions, deletions happen here, there might not be immediate consequences, or maybe we cannot detect. Sometimes we can detect because the regulatory elements or promoter elements are usually affected. And some other mutations can also happen, such as within the open reading frame. Until now, what we have seen is mutations within promoter region, mutations within the intergenic region, and now mutations within the open reading frame. Say the start codon ATG could be mutated to something else, and then we would lose the whole open reading frame. Open reading frame is something, it starts with ATG with continuous codons and stops with the stop codon, or ends with the stop codon. Whereas if in this case, as shown here, if mutation occurred in the ATG region, say for example ATG has become ACG, it is no longer start codon, therefore this is no longer an open reading frame. So those kinds of mutations will not allow a protein to be produced. Although transcribed, there will not be any protein to be produced. Similarly, we may have many mutations within the open reading frame, which we will discuss, but there are also mutations that can happen within the stop codon. A stop codon does not code for any protein, but because of a mutation, it becomes a sense codon. It might code, and the ribosome will keep on translating it until a stop codon, a new one, comes downstream of it, or towards the end, or until the end of the mRNA. So mutations is change in base sequence, but then the consequences will depend on the extent of mutation, the location, and the context of the mutation itself. And there are, at least in point mutations, we can have several of them. What we call them as, say for example, here we have a C, and C is substituted with T. That is, cytosine is a pyrimidine, and thymine is a pyrimidine, and those kinds of mutations are called as transition. If C was replaced or substituted with adenine, which is a purine, then it is called as transversion. Purine to purine or pyrimidine to pyrimidine, it is transition. If it is purine to pyrimidine or the vice versa, then it is termed as transversion. And these kinds of point mutations usually are referred to also as single nucleotide polymorphisms, where only one base, there is a change in substitutions, where different alleles may have different bases in that particular position. Those are referred to as single nucleotide polymorphism, or simply SNP. And we will try to understand a little more details about the consequences of mutations that may occur within the open reading frames. Here I have chosen a small open reading frame, which is ATG and GAC, CAG, GGG, TTG, and TGA. So they code for the respective amino acid that is represented here: methionine, aspartic acid, glutamine, glycine, leucine, and stop codon here. So that we will consider as the normal open reading frame, and it is written in DNA, language of DNA. So there is a mutation here, G and T, and G is a purine and thymine is a pyrimidine, so it is kind of transversion. But that's not what we are discussing. We are trying to describe these mutations in the terms of proteins. So here, this second codon was coding for aspartic acid, but now because it's a TAC, it is coding for tyrosine. And that is, those kinds of mutations are referred to as missense mutations, because they have changed the amino acid from aspartic acid to something else. And if we further see, there is something called as nonsense mutation. That means if there is any mutation, or especially those something like that of substitutions that may have caused premature formation or formation of a premature stop codon, like this, stop codons can be TGA, TAG, and TAA. So here, if the ribosome was translating the complementary mRNA, ATG, and next base, and then there will be a stop codon. So a protein that should have been at least, say, 5 amino acids will now just stop within 2 amino acids. So those kinds of mutations which will allow the formation of stop codon prematurely are referred to as nonsense mutations. Then we have some other mutations, something like this, which are referred to as silent mutations. Say, I hope you remember, or you know that the genetic code is redundant or degenerate, which means that some of the amino acids are coded by more than one codon. Say, for example, in this case, GAC codes for a aspartic acid, and this cytosine in the last one is substituted with thymine. And GAC and GAT still code for the same amino acid, aspartic acid. So although there has been a mutation, there is a change, it's a transition mutation, there is no consequence at the level of protein. That is, it still codes for aspartic acid. Those are referred to as silent mutations. And the cause for silent mutations is because of codon degeneracy. One amino acid can be coded by more than one codon. Not all, but some of them. So then we have something tough or interesting to discuss a little more. ATG, and here we are having a small insertion, CGA. That is, we have added one C, because of which the whole reading frame has changed. Now it is coding for arginine, because the ribosome only reads at one codon at a time, and one codon consists of 3 bases. So here we are now counting it as arginine, CCA as proline, GGG as glycine, etc. Similarly, we can also have deletion. So one of the base, the G was here, which was deleted, because of which now we have a different reading frame. The amino acids are not the same as what the wild type would have had, and there is a whole new amino acid. These codons are new, and they would be coding for different amino acid. I hope it is clear about what is the mutations and these things. I'll try to illustrate a small analogy if it can be of help. Here is a sentence written in English language, all of them three-letter words. Hey, please bin the fly, hug mom, and buy her car. That we will consider as normal sentence to whatever meaning it may be. There was a change in this location, hug, H with B, and now it's something, it reads, hey, please bin the fly, bug mom, and buy her car. But still, it's not the same meaning, but it still carries some of it. Now we have put in 1C in position number 7. So it is hey please, and then there is no meaning to whatever is written here, and that changes everything at all completely. That is something like a frame shift mutation. And you can also have a deletion. Same, the Z here was removed, because of which everything, the meaning of all these words has changed. That is a frame shift mutation caused because of a deletion of one base. There is something we have to remember, and about frame shift mutations is it is deletion or addition of bases, one or more, which are not multiples of 3. That is how we should say. Say in this case, this whole please has been removed. Now let's see, hey bin the fly, hug mom and buy her car. The meaning is carried. Maybe please is not there, except that the rest of the meaning is carried. So the same way, if we are talking about frame shift mutations, and if we have deleted this whole, say for example, this whole CAG, we would still get the rest of the protein. It doesn't mean that the protein is functional, or at the same time it doesn't mean that protein cannot function at all. But what we say, it is not a frame shift mutation. If the reading frame has not been changed, it's not a frame shift mutation. Frame shift mutation will only happen when there is insertion or deletion of more bases which are not multiples of 3. So we could have removed, say, 5 bases, and that would be a frame shift mutation. If you have removed 6 bases, it's not a frame shift mutation. I hope that has cleared some of you or gave much more clarity. Thank you very much. We'll see you in a later talk. Bye.